Welcome to Biology for Bastards, teaching biology in the most profane way you've ever seen or heard. I'm your host, John Doty. Thanks for listening. Back at it after kind of a shit show last week with the dogs. Let's see if this one goes any better. A short and sweet one this time, all about viruses. So this season we're going through the AP Biology curriculum and we're on to chapter 19. So let's just dive right in. So we've talked a bit about bacteria, prokaryotes, all that shit. This week we are talking strictly about viruses. So they are not alive. That's important to, make note of. And they are parasitic, intracellular parasites, not parasites. What the fuck is that? Parasites. And they are very fucking tiny. Very small. We're talking like the size of ribosomes tiny. They're very simple. They're typically just a nucleic acid surrounded by a protein shell called a capsid. The nucleic acid can be DNA or it can be RNA. That RNA can be single-stranded or double-stranded. And then the fanciest viruses have this viral envelope outside the capsid. So it'd be the envelope, then the protein, then the nucleic acid. And they are very quick to reproduce. And because of that, or kind of, not because of it, but alongside with that, they do mutate very easily. And when you have your RNA viruses, they have zero mechanisms for any sort of error checking with their information as it's getting used. So super easy to mutate. And like I said, we could have single-stranded RNA, you can have double-stranded RNA, you can have double-stranded DNA, you can have single-stranded DNA, you can have pretty much, if you can think of a number of strands between 1 and 2, and then DNA or RNA, those viruses exist. Okay, and that's kind of, we've got a nice little table in the PowerPoint if you're following along. Now, when it comes to reproducing, to simplify it a little too much, you have the entry of the virus into the cell. You have the viral components that are replicated. You have the transcription of those viral components, and then the assembly of the new virus as it exits from the cell. So that's any viral replication cycle, but like I said, that's almost too simple. There are two main ways, or two ways that viruses actually reproduce, and it's the lytic cycle and the lysogenic cycle, sometimes also known as the latent cycle. And what the lytic cycle does is it basically infects right away to do everything that I just said. Where it enters, you have replication of the viral components, you have transcription of those components into the viral parts, the viral proteins, and then the assembly of the viruses as they exit the cell. And what happens with those cells is the cells die either through lysis or apoptosis. Now, the lysogenic infection or the lysogenic cycle is a longer one, because what happens is the DNA, the viral DNA, not being very specific there, the viral DNA gets incorporated into the host DNA and gets replicated alongside with it, and then some sort of radiation or chemical factor comes along and initiates the transition from the lysogenic cycle into the lytic cycle. That piece of viral DNA that incorporates into the host DNA. If it's a bacteriophage, we call that little chunk of DNA the prophage. And if it's an animal virus, we call that a provirus. And we know what bacteriophages are. They are just viruses that infect bacterial cells. But in case you forgot, you have that piece of information right there for you. Now, when we're talking about the animal viruses, most of those do have that viral envelope, that viral membrane about the outside of the capsid, which makes it tough for the host to detect the virus, makes it tough for the host to defeat the virus. And then there's another class of viruses out there that are different from the lytic cycle, different from the lysogenic cycle, and those are retroviruses. They are, I guess, most like the lysogenic type of cycle, but in this case you have an RNA virus that uses this enzyme called reverse transcriptase to take the RNA and reverse transcribe it into DNA, and then that DNA is going to insert itself into the host as a provirus. And then as the host transcribes, or as the host replicates, so does the infected cells, so does that provirus, and then eventually the host will transcribe the provirus to make new virus parts. And HIV, human immunodeficiency virus, is a really good example of a retrovirus. There are a bunch of other ones that we are not really going to talk too much about. Stuff like Zika, dengue fever, that chikungunya that I can't pronounce, no matter how much I try. And when it comes to trying to prevent viruses, there's really, vaccines are the best way. Okay, where you get either a weakened virus or part of the virus that will trigger an immune response, so that in the future you can fight off that virus. So that's things like the HPV vaccine for the human papillomavirus, the MMR for the measles, mumps, and rubella, hepatitis A, the flu shot, all of those vaccines against viruses. Now, if you do get an infection, antibiotics don't work because antibiotics target bacteria. You would need an antiviral drug. And what those do is blocks viral replication after an infection actually happens. So there's that. So vaccines are your best bet. There's not too many antivirals out there. You have stuff like AZT for HIV infections. You have stuff like Tamiflu, but if you've ever paid attention to Tamiflu, you've got to start taking that early on. Otherwise, it's not going to work by the time you have too much viral load in your body. Now, we have two last things, and then this episode is short and fucking sweet, because viruses are pretty straightforward. We have viroids, which are small, circular little RNA molecules that infect plants. And really what they do is they mess up plant growth, just regulatory systems that control the plant growth. Just kind of get in the way, start screwing shit up. And then we have prions. I've heard them called prions. I don't know how to pronounce it. Prions. I've heard it both ways, which are these infectious proteins. And these are kind of scary. They cause scrapie in sheep, mad cow disease, or what is it? Bovine spongiform encephalitis, I think is the technical name of mad cow disease. Creutzfeldt-Jakob disease in humans, Kuru in humans that people get from eating dead people, eating the brains of dead people. Yeah. Now, the prions, or the prions, they're kind of scary because there's at least an incubation period of like 10 years. So you can get infected and you will have no absolute clue for like 10 fucking years. And that's crazy. And the prions are almost indestructible. Like we don't know how to fix things. If you have a prion disease, you're fucked. We don't know how to do anything to prevent it, anything to cure it, anything to treat it. We're just, like I said, you're just fucking screwed. And then there are some prion neurodegenerative diseases that, again, we don't have any cure for. We're working on them. Stuff like Alzheimer's and Parkinson's. So this episode, real short, because that's everything. That's viruses for you. Real short, real sweet, kind of on a downer note. So Bastard of the Week is Megan out in Oregon. I don't know how to pronounce that. Never have. Left us a review on iTunes that I just saw. So thanks, Megan. I appreciate it. I've never left a review either, so I don't really know what you had to do that, but I really, really appreciate it. So on that note, I'll wrap stuff up. Our intro and outro music is the song Feeling Good by Purple Planet Music. Follow us on Facebook, Twitter, Instagram. Visit our website, www.biologyforbastards.com. And until next time, thanks for listening.